Good morning, everybody. Hope you all are doing great. Welcome to day 3 of the Montana Brewers Conference. This is Matt with the MBA. And we have Chris Palmer from the Department of Chemistry and Biochemistry at the University of Montana. We're very pleased to have Chris with us here this morning. I'm going to turn it over to Chris, turn my camera off, but before I did that, I just wanted to let everybody know we do have a chat bar on the side. You can enter questions there. If you'd rather ask your question verbally, just indicate on the chat that you have a question and we can bring you on live to ask those questions, and we'll have some time at the end for some Q&A. With that, I will turn it over to Chris.
Okay, thank you, Matt. And also want to thank Ryan for reaching out and inviting me to give a talk here for this conference. What I want to talk about today is some of the plans we have at the University of Montana for some educational programs, but also for an analytical program on campus that we hope will turn into a lot of collaborative relationships, and also we'll be able to provide some services for the local brewing industry. So if you guys see the next slide, please. Just to give a quick overview so you don't get lost in this whole presentation today, I'm going to talk first about the instructional programming proposals. We have a few different programs that we hope to get started next fall on campus for students on campus, but also for people in the community to be able to come to campus, or perhaps even take the course online and gain some of the information and knowledge in the course. And then I will talk a little bit about some of the representative course content, some of the things I hope to cover in these courses. Some of that may be familiar to you. If you're a brewer, you probably already know some of that chemistry or much of that chemistry, but I just want to give everybody an idea of the types of things that we hope to include in our course and in our certificates and in the badges that we'll be offering. And then I'll talk a little bit about the analysis program and the proposal to set up a laboratory here on campus. That laboratory will be for students to intern and to learn, also as part of the course, to learn some of the analytical techniques that are important in terms of understanding what's going on in our beer. And then I hope I am leaving enough time for some feedback. I'd like to get as much, have people ask questions, but also any kind of suggestions or recommendations you have for the types of things that you think that students would most benefit from in a program like this. I'd be happy to hear that. So next slide, please. So I thought I'd spend a little bit of time just introducing myself and talking a little bit about where I come from. I'm actually a PhD in analytical chemistry, and that's chemistry is my first subject, and really I would describe myself as an analytical chemist and not as a brewer or as a brewing scientist, at least not yet. Next year I hope to take a sabbatical and maybe do some studies, some more specific studies in brewing science. But in any case, right now I'm an analytical chemist first, and somebody that has interest in Zymergy and in brewing chemistry. So after I earned my PhD, I did postdocs in Virginia and in the Netherlands and in Japan. And all of those, of course, were focused on analytical chemistry as well. But I guess part of the reason to bring this up is that, during the time I spent in the Netherlands, 18 months that I spent in the Netherlands, I made frequent trips to Belgium, England, Germany, and of course enjoyed a lot of new styles of beer that I had never experienced in the United States, and very much enjoyed that experience, and also developed quite an interest in the brewing and the brewing chemistry, the brewing science. Later in my career, I spent a summer in Univ in Belgium, and really fond memories of sitting outside along the streetways there in Univ, enjoying a Hoegaarten or another Belgian beer, a triple or a double. And so, again, sort of developed an interest in brewing and in good beer during my time in Europe. I took my first academic job in Socorro, New Mexico. That was back in 1994, I guess. And it was shocking, right? Because having lived overseas and having spent a lot of time traveling around in Belgium and England and Germany, this was sort of the beginning of the new craft brewing explosion in the United States, and there wasn't any craft brewing going on in Socorro. There was no craft brewery there. So became pretty involved in home brewing while I lived in Socorro, and brewed a lot of beer in my house. Made a lot of mistakes along the way, but learned a lot from the mistakes that I made. And then something that maybe I would not recommend that an assistant professor do without tenure, but I started a Zymergy club on the campus there at New Mexico Tech. And I say maybe I wouldn't recommend that to other assistant professors because I got in a little bit of trouble when they realized that the Zymergy Club was involved in brewing beer and also drinking the beer that we brewed. And I guess they didn't think that was a proper activity for a faculty member to be engaged in with students, but I continue to disagree with that. And then later, after I'd been there for 4 or 5 years, the microbrewery did open in Socorro. It's called Socorro Springs Brewing, and I was pretty good friends with the person who opened that brewery, started that brewery. He was on the faculty in geosciences at the New Mexico Tech. Okay, and then I moved up here in 2001, so I've been in Missoula since 2001. Sometime in the last 10 or 12 years, I started providing some analytical services for some of the local breweries here in Missoula, primarily ABV content, but we've also done some IBU analysis, and we have plans for some additional analyses that I'm certain that we can get up and running. Did a sabbatical in Tasmania, Australia, which is a place where they grow a lot of hops, and was somewhat surprised to learn that the craft brewing industry was just beginning there in Australia when we were living there in 2007, 2008. So there was only one microbrewery called Moo Brew there in Hobart, Tasmania. So now I've put together this proposal for a certificate and a micro-credential program here at the University of Montana, and that's what I wanted to describe to you today. So next slide, please. So there's 3 basic curriculum proposals. The university system's a bureaucratic system. There's a lot of different levels of proposals and things like that. So there's 3 different proposals that are that have been submitted now. The first is for what we call a Certificate of Applied Science, and this is going to be a full-on science, chemistry, biochemistry, or microbiology type degree. So it's really designed for the audience for this degree would be the natural science majors on campus. So students that are in a 4-year program here on campus that are studying either chemistry or biochemistry or perhaps biology or microbiology, they could, by taking just one additional course, a 3-credit course in brewing science that we're going to be introducing, and that I'll describe to you in a few minutes, by taking just that one course, they'll be able to earn a certificate. That course is going to be very specifically tied to the chemistry and biochemistry and microbiology of brewing. And so they'll gain quite a bit of experience there, but they'll also, I think, see the practical side of their studies. They'll see a lot more practical chemistry, practical microbiology, practical biochemistry in this brewing science course. So it'll be an opportunity for them to see sort of the application of their studies to an area of interest to many students. Alternatively, we're going to have a certificate of completion, which would be a fewer credits involved. And this is really designed, again, for other students on campus, for students in 4-year programs here on campus, perhaps for students in 2-year programs at Missoula College. Thinking about students in programs like wildlife biology or health-related programs, where they only take a little bit of chemistry, a little bit of biology. They don't take the biochemistry and the microbiology courses that the natural science majors would take. And so for them to complete a certificate, what we have in mind is that they would first complete a 1-credit Intro to Brewing Science class, which would provide them with some of the background in chemistry and biochemistry that they need to be able to understand what's going on in the full brewing science class that the other students would be taking. And then the last program here is really for community members, maybe perhaps for brewery employees or for home brewers, people who have just an interest in brewing science, want to become better brewers perhaps. They could sign up for these courses as non-credit courses and earn what we're calling a micro-credential. This is the latest thing in higher education, that people can earn a micro-credential. It comes with a digital badge, which you can keep on with your resume and in LinkedIn and that sort of thing. And so it gives you some sort of credentials. All right, next slide, I guess, please. So the Certificate of Applied Science, we don't need to go through all this in any detail really, but just to say that it will involve a full year of chemistry, and then biology. The principles, sort of the basic biology class, is Bio B 160, but then cellular and molecular biology and general microbiology, as well as a microbiology lab. Students will take a semester of biochemistry. They'll take a semester of quantitative analysis, which is an analytical chemistry course, which will prepare them to be able to do some of the analytical labs that'll be part of the 314, Chemistry 314, which is this brewing science class that we're introducing in the fall. Next slide, please. Whereas the certificate of completion would be lesser background material, as I said, so that people would take 1 year of chemistry and they would take the regular biology class, general biology class, but then through this Introduction to Brewing Science class, they'd be prepared to take Chemistry 314, the brewing science class that we have in mind. And this Chemistry 314 class, I have a lot of things I'd like to be able to do with it. If everything goes through the approvals and if we get a little bit of seed funding, then this will be offered for the first time next fall, in fall of 2001. And we would like to collaborate with local breweries or perhaps with the program at Flathead Valley Community College to produce a batch of beer each year that we teach this Chemistry 314 class. So the students would get not only the lecture component and some understanding of the chemistry, biochemistry, and microbiology behind brewing, but they would also get some practical experience in actually putting together a batch of beer. I've talked to a couple of breweries here in Missoula, and they've expressed some interest in collaborating on a project like this. You could take it as far as you want to take it, I guess. I had thought about trying to do it in such a way that it would be ready for Grizz Cat weekend, and then we could have a local Grizzly produced beer for the big game weekend. But there's a lot of different ways that we could approach something like that. And if any of you have interest in doing something like that, please let me know. We'd be happy to work with you. The other thing is that students who have completed this certificate program are going to be in a really good position to either intern in the analytical laboratory here on campus or with local breweries if there's interest from any local brewers that they would like to have an intern working in their brewery for a period of time, then we should talk about that and we should arrange for that. Next slide, please. So then the micro-credential at this point is going to be a non-credit program. And so the advantage of a non-credit program is that people in the community could easily, I want to say easily, as I said, the university is a bureaucracy, and so there's always troubles and challenges getting enrolled. But if people in the community are interested in getting enrolled, they can enroll as a non-credit student. The fees are less than if you were enrolled as a full-time student and paying full tuition. And you don't receive credit for your transcript like a regular student would, but you could receive a micro-credential and a badge, as I said before. I haven't fully explored the online option yet, but as of right now, and today, right this moment, in fact, I'm learning a lot about how we can have our lectures available online. So it may very well be that next fall, when we're teaching this class, if there's somebody who wants to sign up as a non-credit student, that they could also sign up online and not have to be on campus to actually take the class. Next slide, please. Okay, so I want to spend a little bit of time just going over some of the things that we will cover in this class, and some of the, I don't want to get in too far into the chemistry or biochemistry, but I just want to give an idea, an overview of the types of things that I have in mind for these classes. And so everybody who's participating now is familiar with this list of what's typically found in beer, some malted grain, often barley, and that grain is the source of sugar, enzymes, amino acids, other nutrients that are necessary for the yeast and for fermentation. Water, of course, and water is really very important. The chemistry of the water that's used for making beer can be very important. And so not so much the water itself, but what's dissolved in the water. We want pure water, but we also want water that has appropriate levels of various ions and salts dissolved in the water. And then hops themselves and the chemistry, the resin acids, the essential oils that are part of the hops, and that they provide some bitterness and aroma and also preservative properties for the beer. Very interesting talk yesterday morning by John that talked about sort of the hops and the various resin acids and essential oils that are found in these New England IPAs. And well, I'll come back to that a couple of times probably as we go through the rest of this material. And then we'll talk a lot about yeast, the fact that yeast is a microorganism and that it is responsible for the fermentation that leads to ethanol, but also to additional flavor compounds in our beer. Next slide, please. Next slide, Matt. There we go. And so the basic chemical process that's involved in brewing beer is that we start with the sugar and glucose, but mostly maltose sugar. Yeast uses that sugar as food, basically, and carries out the fermentation process to generate ethanol and carbon dioxide. So that's the basic chemistry that's involved. One of the things that's unique and that we'll talk about in class, in this class, is that yeast is, of course, involved in fermentation, and it's an anaerobic process without oxygen, and it generates sufficient energy for the yeast to survive and thrive in the system. But if you click to the next point here, it should bring up the respiration equation. So most microorganisms, or many microorganisms, or even biological organisms such as you and I, will actually use sugar in a different way to generate energy, and that's respiration. And so respiration requires oxygen, so it's not an anaerobic process. It's a process that utilizes oxygen. But the real advantage of respiration versus fermentation is that it generates about 14 times as much energy for the organism to use for survival, basically, and to grow and to thrive. So yeast actually prefers to utilize fermentation. It is capable of respiration, but as long as there's a significant quantity of sugar around, the yeast will actually stick with fermentation. And certainly when there's no oxygen around, then the yeast is still able to continue through the fermentation process to generate energy and to reproduce itself. Next slide, please. So this is that general chemistry equation again, but in the class, we'll talk about this being a stepwise chemical process that's catalyzed by enzymes, which are proteins found in the yeast. And we'll talk about then that what we really need to produce beer is we need a source of fermentable sugar, and we need active yeast. And that active yeast needs various nutrients, including amino acids, in order to survive in the system. So next slide, please. So the Chemistry 313 class, sort of the Intro to Brewing Science class that I have in mind, would actually review and introduce the chemistry of some of the basic chemistry and biochemistry that's necessary to understand the rest of that brewing process. So, for example, the structure of alpha D glucose, which is shown here. The alpha terminology really refers to the position of that OH. So each of those angles in that ring is a carbon atom, and then each carbon atom is numbered 1, 2, 3, 4, 5, 6, and then attached to position 1 there is a hydroxyl group, an OH group, and that hydroxyl group is on the opposite side of the ring from the hydroxyl group that's attached to position 5 and 6. So that's what we mean by alpha D-glucose, that hydroxyl is in that position. And if you see the next part of this slide, if we attach two glucose molecules, two alpha D-glucose molecules together like this, we get maltose, and that's the primary sugar that's going to be involved for yeast fermentation. And then if you next, please. But that sugar actually shows up in the grain as starch. And the starch is not accessible to the yeast for fermentation. It has to be broken down into maltose first. So the starch along the backbone there is really just alpha D-glucose, a lot of alpha D-glucose molecules attached to one another like this in a long chain. And when we have a single long chain, that's known as amylose. And when we have branched groups, like you see above that long chain, that's called amylopectin, and it's important for a variety of reasons when we start talking about the specifics of fermentation and of beer production. And then next, please, that's the cellulose, and cellulose is actually just glucose also attached together in a long chain, but the difference is that this is now beta glucose, and so that means that you get a much more linear type chain that is able to stack and form very stable structures that are not so very soluble in water and that are not so accessible to the same enzymes that will break down starch. So we have to have separate enzymes to break the cellulose, the beta glucan, and we also have to have, we don't really utilize the cellulose in the as a food source for the yeast. Next slide. So the enzymes, and these are proteins, and we'll talk a little bit about proteins and enzymes in the introductory course, but then we'll talk about the activity of each of these enzymes and the importance of each of these enzymes in more detail when we get into the more detailed course, the actual brewing class. These two enzymes are very, very important enzymes in the process. Alpha amylase and beta amylase, they're involved in converting the starch into maltose, which is available then for the yeast. Alpha amylase has the two little circles that are there. The pale circle is a calcium atom, and the green circle is a chloride atom. So calcium and chloride are both important for amylase to be able to be active. Beta amylase doesn't have those same metal ions or ions associated with it. But then the other thing that's really important for students to understand in this introductory course, but throughout the entire course, is that the three-dimensional structure of these proteins, which is shown here as a sort of colorful cartoon, but that three-dimensional structure is extremely important. If they don't maintain this three-dimensional structure, they will not have their activity, and they will not be able to carry out the processes that are necessary for us. So we would not be able to convert the starch into maltose unless we have both alpha amylase and beta amylase in this structure and active in our mash, basically. And so the important thing to remember is that these three-dimensional structures are not guaranteed. That in fact, as we change the conditions, if we heat this too much, or we add too much of a given solvent, or we have too many ions or something dissolved in solution, then the structure of these proteins can actually change and their activity can go away, and we call that denaturing the protein. So next slide, please. So then we'll talk about in the full-scale course, the brewing science class, we'll talk about each of the separate processes involved in production of beer. So malting, mashing, fermentation, and then final production processes involved in making beer. So next slide, please. So malt, basically what we'll talk about in this class is the chemical and biochemical processes that are going on during the malting process. So this starts, of course, with increasing the moisture content of the grain and allowing it to just begin to ferment. And as it just begins to to germinate, as it just begins to germinate, then what we expect will happen, or we know will happen, is that the embryo will produce and release enzymes, the amylase enzymes that I was just showing you, for example, and these will work their way into the endosperm, where we find the starch is stored for the grain. We want this process to go far enough so that we actually do have some of those enzymes produced and released and that they're available, but we don't want it to go so far that we're actually already breaking down the starch and that the starch is being consumed by the plant as it begins to grow. So we want to free up the starch, but we don't want to actually start to consume the starch in this process. Next slide, please. And then we halt that germination process by heating or kilning the malt. And what this will do is arrest the germination and halt the enzymatic digestion. It'll dry the material, the grain, and the malted grain. And then in addition, depending how hot you make that grain or how high a temperature you take it and for how long, then there's various other chemical processes that take place that convert or that generate flavor compounds and generate color in the malt. But also, as you increase the temperature of the malt to such high temperatures, you will denature a lot of those proteins. So we need those proteins. We still need those proteins. We need those enzymes to be active in the next steps of the brewing process. So a pale malt, for example, will still have active enzymes available for conversion of starch into maltose, whereas a chocolate malt that's been heated to such high temperatures, those enzymes are no longer active. So next slide, please. Some of the chemical processes that are going on, and again, I don't want to get into these kind of details here today, but some of the chemical processes that are going on are the same that go on when we cook any kind of food, and these were worked out by a French chemist, Maillard, and Maillard worked out not exactly, but sort of what the chemistry is, or was able to determine what kind of chemistry is going on here, what kind of compounds are being produced. And these compounds produce various flavors, of course, as well as aromas, and they also generate the color that we find in a lot of the malted grain and kilned malt, excuse me. Next slide. So then we'll move on into the mashing process, and I can see I'm now 30 minutes in, so I'm going to go pretty quickly through some of the rest of these slides and just sort of go over, give you a basic idea what types of things we're going to talk about in the class. But basically the mashing process is where these enzymes are going to break down proteins into amino acids, and also break down the beta glucanase into beta glucans, or into, I'm sorry, smaller molecules, because if we have these large beta glucans, that'll result in really high viscosity systems. Next slide. We'll also talk about how we gelatinize the starch and how that's different for different types of grains. We'll talk about the activity of these enzymes, the amylase, alpha and beta amylase, and that they will break that starch down into maltose. But also, we'll talk about the fact that this branching point, these amylopectin points along that starch molecule, is the amylopectin is not accessible to the yeast for fermentation. And so if those are not broken down, if we don't break those branch points off somehow, then those amylopectin molecules will remain in the sugar, remain in the beer even through the fermentation process. And that results in a couple of things. It results in different mouthfeel and sweetness of the beer, but it also results in higher calorie counts in the beer. So those producers who are working to produce light beer or low calorie beer, they will normally need to add some sort of exogenous enzyme at this point, which is able to break down those amylopectin molecules so that they can then be consumed and they don't show up as calories or sugars in the final product. Okay, next slide, please. So then after the mashing process, you would filter and then bring the wort to a boil and add hops at some point during that process. And we'll talk about in this course the various chemical processes that are taking place during the boil, and also the reasons for having this boil take place as part of the brewing process. And there's a lot of different things that we achieve through this, this stage of the process. So next slide, please. The hops themselves that we're adding at this point. We'll talk a little bit about the history of the hops, but mostly about the chemistry and what kind of chemical compounds are available in the hops and that are utilized in the beer. We'll talk about the alpha acids and the conversion to iso-alpha acids, some what you heard yesterday if you listened to the talk about the New England IPAs. And we'll talk also about different varieties of hops and how they produce different or provide different ratios of some of the aroma oils and compounds like that versus the resins. These photographs here, I need to replace or update the one of my house. Those are hops growing on the side of my house here in Missoula. But the second photograph is actually at Great Burn Brewing here in Missoula. Every year they have they brew a batch of beer they call the Backyard Pale Ale. And basically they have the customers bring their hops that they've grown at home in, and we pick the hops and then they brew a batch of beer using locally grown hops. Okay, next slide, please. So the conversion that we've talked about, or I mentioned briefly, and that John talked about quite a bit yesterday in his presentation, these alpha acids are what are actually exist in the hops. And when we heat the hops in the boiling process, we convert those alpha acids into so-called iso-alpha acids. The iso-alpha acids are much more water soluble. And then also they are much more bitter. The alpha acids have about 1/10 of the bittering properties as the iso-alpha acids. And there's of course other chemistry that can take place and that John talked about yesterday, oxidation of these iso-alpha acids. And so we'll talk about that in some detail in this course as well. Next slide, please. We'll talk about adding the hops during the boil, but also dry hopping later in the process or at the end of the process or even during fermentation, as was described yesterday. And the different types of compounds then that are going to be extracted into the product during the boil, and the conversion processes, of course, they're taking place during the boil, but then for late and dry hopping, those processes are no longer taking place. And we're able to preserve a lot of the oils and aroma compounds that we want to have in beer if we're going through this dry hopping process. Next slide, please. We'll talk about some of the things that can go wrong with beer. Those iso-alpha acids, and when they are in the presence of UV light or even visible light, they can be converted through a photochemical process and ultimately yield this 3-methyl-bute-2-ene-1-thiol compound, which is not the compound found in skunk spray, but it does give a odor and a sense of skunkiness to the beer. And so that's why light struck beer is often referred to as being skunky beer. Next, just show that the best way perhaps to eliminate this process or prevent this process is to protect your beer from light, and that's why it's a good idea to put beer in cans or in brown bottles and not in clear bottles. Next slide, please. Another way that you can go about trying to prevent this skunkiness from taking place, or the light struck beer from taking place, is to stabilize the isoalpha acids. So this is something that's done outside of the brewing process, but you can reduce the isoacids, excuse me, the isoalpha acids using either hydrogen or sodium borohydride or both. And these reduced isoalpha acids actually, or hydrogenated, I should say, isoalpha acids actually are more stable and less likely to be converted through interaction with light. Okay, next slide, please. So then, the following processes, the follow-up processes to filter, cool it down to where you can actually have the fermentation take place, and that will depend whether you're producing a lager or English style beer or ale type beer. Different species of yeast that are involved, of course, in that process. Some breweries will talk about the difference between sales gravity and high gravity in terms of how different breweries might produce beer. Next slide, please. And then we'll talk a lot about the microbiology of yeast and the processes, the biochemical processes that yeast is utilizing to generate the ethanol and some of the flavor compounds. We'll talk a little bit about the history of yeast, and I think one of the interesting things is that people produced beer for a very long time and other fermented beverages for a very long time without ever understanding or realizing that yeast is a living organism. And it wasn't until 1860 that the general population actually began to believe that yeast is truly a living organism. So next slide, please. There's different species of yeast and, of course, different strains of yeast, and these are the different species of yeast are used for different types of beer and at different temperatures, but then different strains of yeast that are used to generate different types of flavor compounds. Next slide, please. So again, this basic chemical process is the fermentation process, and if you next, please. Next again, sorry. But it's a lot more complicated than that basic process that I showed at the beginning there. So basically, we're still starting with sugars. We're still ending up with ethanol product, but there's a lot of other steps along the way. There's several different steps along the way, different enzymes that are involved in these steps, and different materials that are produced, or different products that are produced. And if you click maybe one or two more times, these different products have different capabilities, or add different things to the beer, or even maybe add off flavors to the beer. So the next one, please. Some flowery, fruity notes might be desirable. And next, diacetyl, which is actually produced during the fermentation process, but then is mopped up by the yeast toward the very end of the fermentation process, produces these butterscotch or buttery off notes that nobody wants to have in their beer. So this is one of the things that's important in the brewing chemistry and brewing biochemistry. Part of this is to recognize the types of compounds that are being produced, and also to recognize which of those are desirable, which of those are undesirable, and how to minimize or reduce or try to get past the more undesirable compounds that might be produced. Next slide. So after, you want to continue your fermentation until the diacetyl has been consumed, and then afterwards, there's measurements we make on the beer so that we're able to calculate the ethanol content of the beer. We want to remove the bulk of the yeast from the beer. That yeast that's being removed could be reused, but not too many times. Or if you're in England or in Australia, they produce a product from that spent yeast, either Marmite or Vegemite, which is an acquired taste. Next slide, please. The brewing, of course, the carbon dioxide is being produced during the fermentation process, but that doesn't really provide sufficient carbonation for the beer, and so final product will be pressurized or will have additional carbon dioxide dissolved in it through a pressurization process or through a membrane type process. This is one of the mistakes I made early on in brewing. I started a batch of beer. I was going to bottle condition it to carbonate it, but I had to go to a conference and I had to leave for the conference before the beer was really ready to be bottled. But I went ahead and bottled it anyway and went off to the conference and left a few cases of beer there to explode for my wife while I was gone at the conference. I was a pretty popular guy when I got back. Next slide, please. Okay, so then there's all these properties that we associate with beer that we think are, we don't think, we know are properties that we want to find in beer. We want, a decent foam, decent head on the beer. We will talk about in the class the difference between nitro beers and beers with carbon dioxide, and the fact that nitrogen forms smaller bubbles and a more stable head on the beer. The color of the beer is important, and that comes, of course, from the roasted malt, but it also comes to a certain extent during the boiling process. Clarity and haziness. Many styles of beer, of course, we want to have very clear beer, but there's also styles where we anticipate or we want to have a certain amount of haziness associated with the beer. And as John talked about, the proteins that are responsible for a lot of that haziness can also be carrying along other insoluble compounds, including some of these alpha acids and things like that. And of course, we want to have good flavor in our beer. So next slide. So that brings me back around to what we're talking about doing here at UM, and that is brewers are interested and should be interested in the composition of their beer. And for me, that was a very interesting presentation yesterday on the New England IPAs and some of the differences in composition with the New England IPAs relative to the Northwest IPA style. But these are the sorts of things that people want to be able to measure, and that a small microbrewery concern is probably not going to be in a position to buy the equipment and have the expertise available to carry out all of these types of analyses. So we're interested, of course, in the ethanol content. We're interested in bitterness, IBU, not just the isoalpha acids, but the other hop acids and other hop compounds as were described yesterday. We're interested in the color of the beer, its pH, and we want to make sure that our diacetyl concentration is very, very low in the final product, because otherwise we do get those off flavors that people are not pleased by or happy with. Next slide. So what we have in mind for our laboratory here at the University of Montana is to establish these capabilities. We are already doing percent alcohol volume analysis. We use a gas chromatographic method for that. We've been doing that for numerous years now for some of the microbreweries here in town. There's also, of course, an Anton Par piece of equipment that a person could purchase to do this. We don't necessarily have plans to do that. We can already do the analysis with the GC method, but I have been in contact with the distributor for the Anton Par instrumentation, and it may be that we'll have an opportunity to use some of that instrumentation as well. We can measure IBUs right now using the standard spectrophotometric method that was described yesterday. We can also do HPLC analysis. We have the HPLC equipment available for us. I had a student about 4 or 5 years ago who developed those methods, and well, we haven't been doing that analysis recently, but if people are really interested and curious in the alpha acid and other components that are in their beer, then we could do the same sort of HPLC analysis as what was described yesterday. We can do color, we can do pH. Right now, we've been working on a diacetyl method, but my thought is that the method we're using, which is a distillation method, it's actually very time consuming. It's something that takes quite a bit of time, so we wouldn't be able to turn around results very quickly with that method. So I would much prefer to have a headspace gas chromatographic method, and that requires the institution to purchase a headspace sampler that we could use with our gas chromatographic instrumentation. And then we can do zinc and other metals. We have the atomic absorption instrumentation here that allows us to do those types of analyses. Zinc is an important nutrient for yeast, and so people are curious and interested to know how much zinc they have available. All right, services. The idea behind this program is that we would offer services to the local industry and to people across Montana for a reasonable fee. Up until now, the analyses that we have been doing for breweries have been for a very reasonable fee, which is that they provide us with some beer when we have a department picnic or something like that. But we intend to formalize that now and to have a reasonable fee that will allow us to cover the costs of the analyses. The students in the Chemistry 314 class will complete 2 or 4 laboratory exercises in this laboratory, doing some of these analyses and learn about those processes and those analyses. Those who have completed the certificates of either type might choose to intern in this analytical laboratory and work in the laboratory. And then we're also interested in trying to find some seed funding to purchase the headspace analysis or headspace instrument for the gas chromatograph, and also to do some method development work. Okay, next slide. So these are just sort of acknowledgements of where I got a lot of this information. Charlie Bamforth is a source of a great deal of very interesting information about brewing chemistry and brewing science. And then the last slide is just a question or an opportunity for folks to ask questions.
Great, thanks, Chris. Again, folks can either click on ask a question at the bottom of the screen, type your question in the chat, or just type in, I have a question, and we'll call on you.
I also meant to send to put my email address on this last slide here. If any of you have recommendations for content that you think ought to be part of a certificate program, or if you have interest in collaborating either on the in terms of interns or internships for students, or in terms of submitting samples for analysis, we're here and really be quite pleased to work with you.
Great. I'm gonna pop your email into the chat there.
That'd be great. Thank you.
People can grab it off of there. There you go. chris.palmer@msu.umt.edu. All right. Last call for questions. All righty. Well, thank you, Chris. Thank you for the presentation. Thank you for your time. This has been awesome. And
thank you for the opportunity.
And thank you, everybody, for joining this morning.
All right.
All right. And a reminder to everybody, I'm going to take the opportunity to just remind folks we have the virtual pub trivia this afternoon at 4 p.m. Oh, Hannah is just asking, is that she said at msu.umt.edu, and I don't think that's right. It's at mso.umt.edu, right? Yep.
Yes.
Chris, great. So yes, 4 o'clock today, 4 to 5:30, we're doing virtual happy hour, our virtual pub trivia happy hour. So everybody who's registered for the conference should have received that information. If you're looking for an excuse to cut out a little early and grab a beer and do some networking. It should be really fun. The way we're going to set that up, folks will get randomly assigned a team. So presumably you'll get assigned to some folks you maybe don't know so well. Great opportunity to meet some new people and have some fun. So we're not able to get together in person this year. We always have a lot of fun with our social activities around the conference. So we're doing this to try to fill that void. So I hope we have some good turnout this afternoon for that. All righty. Well, thank you, everybody. And thanks again to all of our sponsors, Gallatin Valley Malt, McDanton, Montana Craft Malt, Crosby Hop Farm, Montana Press Monthly, Malt Europe, Whipfley, Arrive, Breeze, Glacier Hops Ranch, JCCS, Anderson's or Malin, First West, All Things Trivia, who's doing the trivia session this afternoon, and Montana Wildlife Federation. All righty. Thanks, everybody. Have a great day.